let's jump right into this explainer on next-generation sequencing. We're talking about a technology that literally revolutionized how we decode the building blocks of life. We went from spending decades trying to decipher just one single genome to being able to sequence thousands in a matter of days. It's wild. Today, we're going to break down the history, the mechanics, and the absolutely incredible future of genomic analysis. To really get why this tech is such a big deal, we gotta rewind to 2001. That's when the original Human Genome Project finally crossed the finish line. And look, it was a massive win for science, but it came with a jaw-dropping reality check. It took over 10 years of grueling work and a cool $3 billion to pull it off. Yep, $3 billion for just one human genome. So obviously, if we ever wanted to use genetics in everyday medicine, we desperately needed a faster, cheaper way to do this. This whole journey actually kicks off back in 1953 with Watson and Crick's famous discovery of the DNA double helix. Then we jump to 1977, which brought us the Sanger sequencing method. But notice that huge gap? Sanger basically ran the show for nearly 30 years, right up until we finished the human genome around 2000. It wasn't until 2005 that everything completely changed. That was the birth of the first next-generation sequencing, or NGS, systems. That year was the true start of massively parallel genomic analysis. So, for 3 decades, Sanger sequencing was basically our only option. It used this chain termination method. Scientists would set up a reaction where these special fluorescent dideoxynucleotides would attach to the growing DNA chain and just stop it in its tracks. By doing this over and over, they could read the base pairs one by one as they went past a detector. Now, it was incredibly accurate, sure, but man, was it slow. And that slowness created a massive bottleneck. Older Sanger machines could generally handle about 96 reads at a time. Just 96. The scientific community was practically begging for a way to process more data without breaking the bank. And that exact hunger is what gave us next-generation sequencing. We went from those 96 slow reads to processing literally millions of reads at the exact same time. What used to take years slashed to just 4 or 5 days, and the cost dropped to roughly 1/6 of the old methods. A total game changer. Okay, before we get lost in the weeds of how NGS works, let's quickly get our terminology straight. First up, reads. When we say reads, we're just talking about the exact sequence of a single tiny piece of our fragmented genomic DNA library. Think of them as the digitized puzzle pieces we create, mostly because we just can't sequence an entire giant chromosome end-to-end all at once. Next is coverage depth. This is basically our quality control. It's the number of times we overlap those independent reads on the exact same spot in the genome. By stacking these reads on top of each other, we can filter out any random mistakes the machine might make, which gives us a super accurate picture of the sequence. And finally, the reference. We need a master scaffold to build on, right? Something like the GRCH38, maintained by the Genome Reference Consortium. We take all our millions of short, new reads and map them against this known master blueprint. It tells us exactly where our data fits and, more importantly, where the mutations are. All right, let's look at the actual workflow. How does this happen in a real lab? It all starts with sample preparation. You don't just toss a drop of blood into a magical machine. First, you have to physically shear or break apart the genomic DNA into smaller fragments. Second, you repair the ends of those fragments to make them nice and blunt. Step 3 is adding special DNA tags, we call them adapters, to those ends. And step 4, you purify the whole sample to wash away the excess chemicals. What's actually happening at the molecular level here is pretty fascinating. Those long strands of genomic DNA are randomly fragmented, and then we ligate, or attach, standardized adapters onto both ends. You can think of these adapters as universal molecular handles. See, the sequencer has no clue what the random internal sequence of your DNA fragment actually is. So it uses those standard handles to grab onto the DNA, anchor it down, and get it primed for reading. Once it's prepped, we need to amplify it to boost the signal so the machine's optics can actually see it. And different platforms have totally different ways of doing this. Some use emulsion PCR. This is wild. They isolate single DNA molecules onto tiny beads inside microscopic water droplets suspended in oil. It literally creates millions of tiny individual test tubes. Other platforms use something called solid-phase bridge amplification, where the DNA actually bends over on a flat glass surface to form little clusters that are super easy for the machine to read. Now, getting the data out of the machine is really just half the battle. Once you've got millions of short DNA sequences sitting on a hard drive, you have to actually make sense of them.
First, we align all those reads to our reference genome. Next, we double-check the read density, or coverage depth, to make sure the quality is solid. And then comes characterization. This is where we actively hunt down the variants. Where does our sample differ from the standard? And exactly what kind of mutation are we looking at?
Because handling this mountain of data is definitely tricky, especially when you hit repetitive parts of the human genome that look identical. It's like trying to put together a puzzle where half the pieces are just the exact same blue sky. To fix this, scientists are constantly tweaking the chemistry to get longer individual reads. Another neat trick is paired end sequencing, where they read both ends of a single fragment. Knowing the exact physical distance between those two ends helps assemble that genetic puzzle with way higher accuracy.
So say we identify all the variants in our sample. We're looking at thousands of differences. How do we know which ones actually matter? Well, we take our findings and cross-reference them with massive global databases, like DBSNP or the 1000 Genomes Project. By comparing our single sample against what we already know about the whole human population, researchers can pluck out those super rare mutations and figure out what a specific gene does or if it's tied to a complex disease.
You might be wondering, why does this whole complex workflow even matter? Simply put, defining the variability in human genomes puts biomedical research into overdrive. It helps us uncover exactly how rare alleles trigger inherited diseases and lets us dive deep into things like epigenomics and non-coding RNA. And it's not just humans. The sheer power of NGS lets us sequence massive, complex genomes, like essential agricultural crop plants, which is a huge deal for global food security. But look, it's not totally perfect. Despite this massive leap forward, we still have hurdles to clear. The industry is always fighting to increase read lengths. We also run into trouble with homopolymer regions. That's when you have a long stretch of the exact same base pair, like 10 A's in a row. Some techs still struggle to count those accurately. And then there's the cost of the infrastructure. Processing this much data requires incredibly expensive supercomputers. We are constantly trying to push that total cost down to the holy grail, an affordable $1,000 personal genome.
Looking ahead, though, the horizon is incredibly exciting. Companies like Pacific Biosciences and Oxford Nanopore are leading the charge on something called single-molecule sequencing. This next generation tech completely skips that whole complex library amplification step we talked about. By reading single DNA molecules directly as they slide through microscopic pores, we eliminate all the biases introduced by PCR. It promises completely unamplified, ultra-long, native genetic reads. Absolutely incredible.
So as we get better at processing millions of reads faster, cheaper, and way more accurately than ever, the possibilities are just staggering. We're shifting away from one single standard reference genome and moving toward deeply personal, highly detailed reference profiles for everyone. Which leads to the ultimate question: will these personalized profiles finally be the key that unlocks true personalized medicine, allowing us to tailor treatments perfectly to your specific, unique genetic code? It's a huge question, and next-generation sequencing is bringing the answer closer every single day.